So I changed the title a bit because, you know, we are in Amsterdam and with many colleagues also at UCL working very hard towards what we call a multiscale model for vascular physiology and pathology, so vascular disease. And you know, the fact that we build multiscale models in this arena, that's what we want to do in VECMA, to put error bars on that to do on surgical quantification. So I wanted to broaden up a little bit and just tell me when my 15 minutes are done and then I stop, because you know this can go on very long, and that's not the idea. So first, this beautiful movie, of course, thanks to our friends at the Barcelona Supercomputing Center, who this was made in the Biomed project, and Peter will show probably more of this beautiful stuff. I think this is a science turned into art, so I keep showing this. Here you see basically a simulation of red blood cells as they move, and then artists put a beautiful rendering around it to show a little bit, let's say a wound of an artery, and this is where hemostasis, stopping the healing of that wound should start. And one of the things that we want to deeper and better understand are the very initial stages of thrombosis and hemostasis. But it's still an unsolved problem, and we believe that this kind of very detailed modeling can help there. And I won't have time to go into that, but it's maybe for a next meeting. So, if you look at vascular models, these are basically the scales that we want to cover. We want to go from whole body modeling. We can do these things now pretty standard using so-called one-dimensional models, and you find it here. So that's on let's say meter length scales, and I know the UCL team is working on actually doing full-blown 3D simulations on these kinds of structures, which would be pretty exciting because then we can finally validate this 1D in these whole arterial trees. We're still not sure about the validity of that because we need to always, you know, struggle with boundary conditions of these things. Anyway, as you go down the scale, we get into the field that many of us started in, three-dimensional hemodynamics. In other words, solving Navier-Stokes equations in some complex geometry. We all like that. We all can do that with our favorite methods. Lattice bolts, Navier-Stokes, DPG, what have you. Then you go down, and let's say this starts at the millimeter range, down to the micrometer range. You start to actually see biology. You start to see living cells, and they interact with each other, and they do things. And this is where the cell-based blood flow modeling would fit in this box. And then we can even go subcell and see what molecules do. And one protein that I find extremely interesting is the von Willebrand factor. This is an interesting protein because it plays a pivotal role in this initial aggregation of platelets. It's a funny one. Did you know that the von Willebrand can be as long as 0.5 millimeter? It's completely unclosed. It's unbelievable. So I would like to understand a bit better how we could use coarse-grained MD to do something with that. Anyway, that's for over dinner. Of course, what we want is to glue all this stuff together, and the type of applications that we would like to do in VECMA are a little bit here, coupling cellular to 3D two-way. So that means they're tightly coupled, and there is a fast dynamics that is tightly coupled to the slow dynamics, and that's in terms of uncertainty quantification, that's where the trouble starts, and that's where also where Anna will show some first results in there. Okay. So this virtual artery, therefore, is a multiscale model for peripheral blood flow. It should, in the end, include all relevant biological, biophysical, and biochemical processes on all scales. Of course, that's like, you know, building a model of everything. This is, of course, nonsense. We take out bits and pieces and then we apply it to challenging problems. We want to study fundamental curiosity-driven questions. What about this initial aggregation of platelets? Why does a thrombus have the porosity that we find in certain parts in the body? This is all not understood. But you could also make it much more applied in the sense that we want to improve treatments or medical devices, or we want to apply all this in silico clinical trials. And you know, we are running many projects in Amsterdam where we focus on either of these units. And then you can even go deeper and go into personalized medicine. This is not a place to discuss this, but we're quite active in that arena. So what is this, this virtual artery? I will quickly go through this. We take this whole body, one-dimensional modeling. We couple that to 3 or 4D hemodynamics. So for instance, here we would be interested in some flow in the brain. And then we couple that to cell-based models. And so we could open up a little part here in an artery and then look at the individual red blood cells and platelets as they flow. Or look at the individual cells. I think I have another picture. There are many pictures here. Or look at individual cells as they make of the arterial wall and look at the biological response to all kinds of stimuli. This is where, in terms of uncertainty quantification, many problems also start, because the kind of data you get from our friends in the biology lab are pretty messy. And sometimes you get experimental data like, if we kick this protein, then something happens there. And that's the best we get. You try to turn that into some rule-based model or something that allows us to compute. Anyway, again, the kind of applications we would like to do in VECMA are a little bit in this sphere. Here you see a picture of an in-stent restenosis, an old problem that we have been looking at for a long time now, but we actually have this stuff validated now against real histology, and we can apply this to the human case. And in the so-called InSilk project, we are now building in silico clinical trials to design stents. So very much an applied project, but for that we need error bars. We need to put error bars on these simulations, and this is where VECMA comes in, obviously. Another one is more on the level of the curiosity-driven stuff, where we look at our cell-based blood flow simulations, and this is typically a hard problem also for VECMA in terms of the compute. And I will say a few words about that in a minute. But that's more, we have so many free parameters in these models, and it is not so clear how all the errors propagate through the system, let alone if we have picked a good model at all, and we have to compare that in a good statistical way against real data. Okay, so this is how we hope in the end it will look like, right? And Peter is also working towards this. And it would be great to have, you know, at some point all these couplings ready, and we're slowly working in that direction. So we have, and now I get to the cell-based blood flow modeling part. We have a code which is called Hemocell, which is short for high performance cellular framework. This is state of the art. We can do so-called high hematocrit flows, meaning high concentration of red blood cells as you find them in human blood, up to 40%. Many codes claim that they can do this, but they can up to 30%, and then the whole thing breaks down. We have put lots of time and effort to push this. We run this on big machines. We're currently upscaling this to Mare Nostrum. I just heard that we got some nice money for one of our programmers to go to Barcelona and actually go to the next level with all this. It's fully parallelized, you have seen this. The main point is this is running in production, and so this is a fast track application for VECMA. We can now take it, glue it to something else, and actually run UQ on this. And I would be very, very eager to start trying this. If you're interested in this, it's freely available. This is the website. Just go there, download it, and through another beautiful project that Peter is also running, we can actually offer support to use this. This is great.
5 more minutes.
Yes, I'm almost done. Thank you. This is an example of using Hemocell for thrombosis, and just to show you a little bit, you know, the troubles that we hit, and you know, you may want to start to think with me how we could do UQ in these kinds of environments. I don't know the answer. I hope by the end of this project to get a little bit my head around this. And so basically what we're after is, and you have a little wound in an artery, and this is a nice sketch, of course. You know, many biological things happen. Lots of this stuff is not in my models at all, right? And for instance, we believe that when the endothelium is a little bit is a little bit damaged down here, that there is a kind of sympathetic nerve response which happens on the level of minutes, where you get so-called vasoconstriction. So you get this little closing of the vessel. This makes sense, because if you're bleeding, you want to close. But this also generates an interesting hemodynamical environment exactly where platelets should be. So this response could also be very beneficial for the transport of platelets to the site of the thrombosis. We're exploring this, and this is an hypothesis. And I really would like to put, numbers on the quality of this hypothesis. So I need air bars there as well. Okay. So this is the kind of data we're working with. I hope the movie starts. So what you see here is a movie of an in vitro experiment where you have a severe stenosis, and this surface here is coated with von Willebrand molecules, and this is being perfused with whole blood at very high shear stresses. And what we find is initial aggregates of platelets here on this side. This is the kind of data we have. We also have this kind of data where you have small spheres that we put on the surface, not we, our experimental friends, put on the surface of a microfluidic device. Again, the spheres are coated with von Willebrands. You perfuse the whole thing with blood. And then after 2 minutes, you find platelets here. So what is going on? We would like to understand this. And what we believe happens is the following. Maybe there is a better, this is another movie, I'll show that later. This is a nice top view. What we believe is going on, and we're writing on the paper right now. If you would do a continuous hemodynamic simulation, you find things. But then you completely miss the cellular nature of blood. And the interesting thing is, if you run the Hemocell simulation, you start to paint another picture of this process. We see behind this big sphere a cell-free volume appearing. At the bottom, this simulation is not completely right. We have better results now. At the bottom, the cells will also be lifted away because of cell margination. So you get an environment where platelets could actually go into this region behind the sphere. The shear stresses are also such that von Willebrand can uncoil. And the final ingredient is that the platelets stay long enough in that region so that it can react with the von Willebrand. So there are 3 factors, and we really would like to pinpoint that. And our only observation is that after 2 minutes you find this. Yeah, so, I mean, I don't know if I can even do a U, or if it would be sensible at all to do UQ on this kind of stuff. I mean, what I can compute probably would be a probability that under my hypothesis I find something like this. Or maybe I should go back to the experimentalists first and see what we can do on this data. Right, well, this is one of the applications that we have in the back of our head. Probably the incident restenosis that I didn't talk about is the first one to jump on, because that's the one we want to put in that insert project. I think this is it. Oh, this is another experiment. I mean, this is all beautiful stuff, but not for now. So wrapping up, we have Hemocell available. I showed you a few examples where we use it. We apply it in many different fields, and depending on what happens in the next year, we will pick the one that I believe will best fit VECMA. For instance, here we're looking at late stages of malaria infections, which is an interesting one. We're looking at thrombosis, microcirculation, properties of white blood cells, etc. So I would say, driven by experimental data, curiosity-driven questions, and clinical applications, we continue to develop the virtual artery, validated against data that we have. And then, where possible, do a VVUQ, where all the V's are important. And since, we would like to follow the FDA, the Food and Drug Administration in the US, who have been writing down very clear procedures to do a VVUQ on biomedical applications and software, I would like to bring that into VECMA to follow that procedure and take one of these examples, or maybe from UCL, to actually implement it according to the rules of FDA. That would be really a thing that would have, or would create impact. That's it. Thank you.
Thanks.
Yeah, that's it.
Alphonse.